Thank you for attending my talk. Today I'd like to present our new finding. Host restriction factor MxB sequesters HIV-RAV protein in the cytoplasm and inhibits RAV-dependent viral gag expression. First, I'd like to briefly introduce MxB protein. MxB belongs to the dynamic-like large GTPase family and is an interval-inducible protein. In 2013, MxB was first reported to inhibit HIV-1 by our group and other two groups. MxB was subsequently shown to restrict herpes virus, HBV, and HCV. The mechanism of MxB inhibiting these viruses has not been fully clarified. One known mechanism is that MxB blocks the nuclear import of HIV-1 DNA and herpes virus DNA. Since MxB blocks HIV-1 DNA nuclear import, we wondered if MxB can also inhibit the nuclear import of viral proteins as part of its antiviral mechanism. To test this hypothesis, we examined the effect of MxB on the subcellular localization of four HIV proteins, integrase, VPR, TIT, and RAV, that are known to localize in the nucleus. This column shows MxB, and this column showed HIV proteins. We can see that in the presence of MxB, integrase, VPR, and TIT still localize in the nucleus. However, RAV changed its subcellular localization from the nucleus to the cytoplasm. These results suggest that MxB causes cytoplasmic retention of HIV-RAV protein. We next try to determine which function of MxB is required to return RAV in the cytoplasm. This is the domain structure of MxB. N-terminal is critical for MxB antiviral function. The first 25 amino acids deleted MxB lose its anti-HIV-1 activity. K131M mutation and is defective in GTP binding. R455D and M574D are defective in MxB oligomerization. We co-transfected RAV and MxB expression plasmid into HeLa cells and examined the RAV subcellular localization. We found that only the first 25 amino acids deleted MxB lose the ability to return RAV in the cytoplasm, suggesting the key role of the first 25 amino acids in MxB inhibition of RAV nuclear import. The function of RAV is to export the HIV RNA, including viral gag RNA, into the cytoplasm so that viral gag can express. Since MxB sequesters RAV within the cytoplasm, we ask whether MxB decreases RAV-mediated expression of HIV gag protein. We used RAV-dependent and RAV-independent gag expression vector and co-transfected either of them with MxB. Western blot results show that MxB significantly decreased RAV-dependent gag expression, but it didn't affect RAV-independent gag expression. This strong inhibition on RAV-dependent gag expression is further confirmed by the profound reduction of viral-like particles, which formed by gag and gag pore proteins. This result demonstrates that MxB inhibits the function of RAV in mediating gag expression. Next, we ask how MxB blocks RAV in the cytoplasm. Transportin 1 is a nuclear importing, have been shown to direct the nuclear import of RAV. We thus performed the PLA assay to measure the association of RAV with transportin 1 and the effect of MxB on this interaction. The right signal reports the interactions between RAV and transportin 1. In MxB expressing cells, there are far fewer red signals, indicating a dramatic decrease in the interaction between RAV and transportin 1, demonstrating that MxB disrupts the association of RAV with transportin 1, thus impairs RAV nuclear import. We next ask whether there are any RAV mutations that resist MxB as the viral countermeasures of MxB inhibition. To answer this question, we started by testing a group of RAV mutations that have been previously characterized to affect RAV multimerization and RAV's function in HIV gene expression. We expressed these RAV mutations with MxB in HeLa cells. The green signal is MxB and the red signal is RAV. We found that L60R and P31A still localize in the nucleus in the presence of MxB, thus resisting MxB inhibition. We also measured the function of these RAV mutations and found that L60R mutant was severely impaired in stimulating GAG expression. We thus focused on P31A mutation in the following studies. We inspected RAV sequences in the HIV-1 strains from HIV patients and found a high degree of polymorphism at position P31. We thus tested these RAV variants at the position P31 for their sensitivity to MxB inhibition. These are the imaging data, and this graph shows a summary of RAV localization with and without MxB expression. We can see that in MxB expressing cells, P31T, P31R, and P31H still localize in the nucleus, thus resistant to MxB inhibition. This result suggests that in HIV strains from patients, there exist RAV variants that resist MxB inhibition of nuclear localization. Although these RAV variants showed resistance to MxB, we noticed that the frequency of these mutations in circulating strains is fairly low. It suggests that besides RAV mutations, HIV-1 may have other mechanisms to overcome this MxB inhibition. Indeed, we found another viral countermeasure. In this experiment, we infected the control and MxB expressing CD4 positive sub-T1 cells with the N-alpha-3C88 virus. We quantified unspliced, singly spliced, and multiply spliced viral RA species in control and MxB expressing cells. We observed a significant increase of multiple spliced RA in MxB expressing cells. Since multiply spliced RA express tight, RAV, and NAF, we performed Western blot to further confirm the dramatic increase of HIV tight, RAV, and NAF expression in the presence of MxB. Based on this data, we propose a model. When full-length viral RNA is synthesized, it undergoes multiple splicing and is delivered to the cytoplasm to produce RAV and tight proteins. Tight comes back to the nucleus to stimulate HIV transcription, and the RAV export unspliced and singly spliced viral RNA into the cytoplasm. In the presence of MxB, RAV cannot get back to the nucleus. The returned unspliced and partial spliced viral RNA species are further spliced to produce more RAV and tight until a high enough level of RAV is produced and surfaced to overcome MxB inhibition. Taken together, we have found that MxB blocks the nuclear import of HIV-1 RAV protein by disrupting the interaction of RAV with transporting 1. HIV-1 have two mechanisms to overcome this inhibition. The first is to acquire MxB-resistant RAV mutations. Second is to produce more RAV protein to overcome MxB inhibition due to the feedback loop of HIV gene expression. This study was published in Emergency Microbes and Infections. This work is supervised by Dr. Chen Liang through collaboration with Dr. Shan Chen, Dr. Fei Guo, and Dr. Wentao Qiao's lab, and supported by funding from CRHR. I also want to thank all the lab members for their help. Thank you very much for your attention.